Hey, now when I get to say those damn biologists and they're naming shit after people all the time, I have somebody that I can look at. Okay, so the two phases, we were talking about the hydrolysis, the chymotrypsin. This is going to involve the covalent catalysis. Remember, that's where we covalently attach to our substrate first. The two phases are actually going to be explained by the formation of your covalent enzyme substrate intermediate. Remember that intermediate is actually going to involve that binding, and that will be a reaction in and of itself, right? And then we'll have the next reaction, which will be product formation, okay? So one molecule of the p-nitrophenolate is going to be produced rapidly as the acyl enzyme intermediate is formed. Again, this is an acyl group. You may have remembered that from organic chemistry, i.e. acylation. See, that's the acylation reaction. We're attaching to the acyl group. The acyl group is the one where we attach a carbon, a bond to a carbon that has a carbonyl on it, and then a carbon chain going off of that, okay? So that's the acyl. You will recognize that by the time we're done with this semester. A lot, okay? This is going to show up in citric acid cycle, and you'll really love that one. So anyways, this is the acylation reaction. We'll get the acylated enzyme, and so this is the acyl enzyme complex, or another way of saying that is the acyl form of the enzyme. We will then have what's called deacylation that will happen to end this reaction. So the deacylation will be where we add water across this to lyse that bond, freeing our enzyme and typically our product. Okay? Though this same mechanism is used in many, many places, that is a truism of biochemistry. If you could use the mechanism once, your biological systems are going to use it 100 times. Okay? Because obviously, if you develop something that works for an enzyme, a catalyst, you're going to reuse it over and over, right? Just targeting different places. So right here we have the chymotrypsin again. These again are these disulfide bonds that we had discussed. And then this is the serine that's going to do that reaction that we had just shown and discussed. Again, that's that serine right there. And so again, the chymotrypsin is going to be roughly spherical. It's going to contain 3 polypeptide chains. These chains were linked by disulfide bonds. Those are the disulfide bonds right there and there. This is that serine again. That's the 195. That's the active site of the chymotrypsin. Again, an active site typically has to lie in some sort of cleft in the surface of your enzyme because you have to gain access to it, right? In some way, shape, or form. Okay, so this is referred to in chymotrypsin. We have what's referred to as the catalytic triad, okay? So this is a set of 3 amino acid residues in the active sites of some enzymes. Read some enzymes as a lot of enzymes. It's actually a very common motif, okay? In chymotrypsin, the side chain SER195 is going to be the hydrogen bonded to an imidazole ring, which is His57. You can actually see that right here. Can you see that? And how it's close proximity, so you can make hydrogen bonds to it. Yeah, you could have come up with all this stuff yourself, right? You don't need me. Dr. Clausen. All right, so the NH group of the His57 imidazole ring is actually going to be hydrogen bonded to the carboxylate group of your ASP102. And again, we can see that over here. Everybody see that okay? Okay. And that's actually your catalytic triad. Again, notice that this is in this little cleft here, and so that is your active site right there, isn't it? All right, and so the His57 position of the SER195 side chain, and so the His57 is going to positions the SER195 side chain and polarizes the serine's hydroxyl group. Remember, if you're making a hydrogen bond, that means you're pulling electron density, right? And so this is going to help polarize that. It'll make it more reactive. So in the presence of your substrate, the His57 will act like a general base catalyst. Remember where we talked about oscillations in the presence of a base? Hey, that organic chemistry is showing up again, isn't it? It's good. So this removes a protein from the Ser 195, a proton, Freudian slip, right? A proton from the Ser 195, it's going to generate a highly reactive alkoxide ion. And so this is actually the same as that base catalyzed acylation reaction we had done so many times when we were talking about like SN2 reactions. We had done a bunch of those, right? This is just that. We're just keeping the base right there so it can help abstract that proton, okay? Instead of putting a strong base in, like an OH minus. The ASP102 is going to then orient the His57. This is going to render it as a better proton acceptor. This does this through the hydrogen bonding and electrostatic effects, i.e. pulling electron density away, right? And donating electron density when it needs it. So it'll help to modulate that electron density. Another way you can think of this is it gives its resonance forms, right? It allows it to spread the electron density or the lack of across more structures, okay? Thus stabilizing the bound form or the alkoxide form in this case. So this right here is what it looks like. There's the ASP102. This is stabilizing this group right here, the His57, through this hydrogen bonding here, okay? And notice again, that's hydrogen bonding with a negative group, right? And so it can share some of that negative charge over, okay? And so this His57 then is going to act as our base. This is actually going to help abstract this hydrogen off of an OH. This will actually form a alkoxide intermediate, okay? This is the higher energy form. This probably exists less in solution in any given physiological pH state. As you remember, alcohols are not particularly great bases normally, but when we have a structure like this, they become much better bases, right? Okay. So now we've formed this alkoxide ion. When we formed the alkoxide ions before, they were actually very good at attacking an SN2 style reaction, right? And so we can have an SN2 style attack that will happen on our carbonyl, thus forming our acyl group, right? Okay, and so here we actually have the tetrahedral the catalytic triad doing the reaction as we had just discussed. Note right here is our protein, okay? And here's our carbonyl in the amide bond, right? Okay, and so what's going to happen is, again, this right here has been stabilized, so it's much better at being an alkoxide than it normally would be, right? Or another way of saying that is it's much more reactive and it has a lot more negative charge it can give into a bond, okay? And so it's actually going to attack the carbonyl, just like we had discussed when we did the chapter on the carboxylate-like substances. One of them was the amide, right? It's just attacking the amide, just like we had talked about then, okay? And so we attack that carbonyl. It's a very SN2 style reaction. Calling this an SN2 is actually pretty accurate, okay? But there's some, know that it might confuse people if you do, okay? You have to kind of talk about it then. So you attack this carbonyl. This is going to cause the electrons to go up onto the carbonyl oxygen. And normally, this is what normally happens in reactions, right? We have that carbonyl double bond go first because they're delocalized electrons, if you remember from last semester. And then they usually came down and caused the reaction to undo, right? Or something else to come off, okay? In this case, we actually have one more part of this active site that we need to talk about, and that's the oxyanion hole. So we actually have something here that will bond to and stabilize this negative charge on this alkoxide right here, or our tetrahedral intermediate. Do you remember the tetrahedral intermediate from last time? Kinda? Okay, so when these electrons come up and you have this extra oxygen bond to this is a tetrahedral structure, right? And so this is the tetrahedral intermediate. It's always formed when you do an attack on a carbonyl because the electrons in the double bond are delocalized, it can come up on the oxygen, right? That's the first thing that happens. Normally, this falls apart really fast, right? Boom. And then we go to either back to reactants or products, okay? In this case, though, we've actually stabilized this. What that means is just this one action has already given us a lower energy activation energy, right? Because our intermediate is now lower energy and more stable, okay? Okay, so we make the tetrahedral intermediate, which we would normally make in this reaction, but we have an oxyanion hole on this enzyme that's going to stabilize it, okay? Now, the next part that's going to happen is we have two things that can happen. We can either release our substrate back in and the complex falls apart. We talked about that, right? That's that K-1 going backwards, okay? The other option is the K2, right? Going forwards in the reaction and making our product, okay? And that would be going this direction right here. So what we're going to see is that this bond here, the amide bond, is actually going to break. This is going to give its electrons into this bond with the hydrogen. Note that that hydrogen is a positive proton that is attached to the base, right? Okay. Or another way of saying that, this is the conjugated acid right now, okay? Okay. This acidity and basicity of this group is highly modulated because we have this complex formed with this hydrogen bonding, giving this kind of wonderful intermediate basicity acidity, okay? That is kind of just right for these reactions. And that's the magic of this enzyme, right? Or the catalytic triad, I should say, because there's multiple enzymes that do this. Okay, so now we've actually attached this nitrogen to the hydrogen here. We have broken the bond between the nitrogen and carbon. Our mid-linkage is snapped, right? We've now cleaved our protein, haven't we? Okay? When we break this bond, the electrons in this oxygen here, the alkoxide here, are going to come back down and reestablish our carbonyl bond. Because otherwise we would have a carbocation, right? And that would not be very stable. Okay, notice, by the way, that this is actually back to a hydrogen bond because we actually moved that electron density that was in this bond onto this hydrogen to form a bond. And then the electron density in this right here has moved back into this ring structure. Okay? Okay, this is the acyl enzyme linkage right here. We have just the acyl group here. We don't have a full protein anymore, right? We've actually broken our protein bond. Okay, or our peptide bond if you want to be more biology-like. Okay, so the next thing that's going to happen here is we're going to lose the amine portion of this, okay? And that will leave us just with the acyl-linked enzyme. So again, we'll keep the acyl-linked enzyme, but we remove the amine portion of the polypeptide bond that was there, the protein, okay? All right, so the next thing we have to do is we now have to release this acyl. This came off first. Can anybody tell me why? Why would the amine come off first? What sort of linkage is this? It's a hydrogen bond, right? Exactly. This is going to be breaking all the time at room temperature or physiological temperatures, right? And so it's going to break all the time and it's going to drift off very easily. Okay? And so the next one, you actually have to do a reaction to break this one, don't you? And so the activation energy of this is much larger, and it's going to take a lot more time, isn't it? Okay? Okay, so the next one is actually going to be a hydrolysis reaction. What we're going to do is we're going to hydrolyze this bond right here between the carbon and oxygen. However, the mechanism of this is kind of interesting because, again, our catalytic triad has that basicity to it, right? Meaning that the water here can actually donate a proton onto this nitrogen. This will make this a much more reactive species, very similar to our base catalyzed hydrolysis. It's just we have a very special base in this case, right? Okay. So again, we're going to donate the proton. This is going to generate locally something that's very like a hydroxide ion, okay? And in fact, it will eventually be a hydroxide ion as you transfer it, okay? But you don't really form free-floating hydroxide, just understand that, Does that make sense? Okay. Okay, so what's going to happen then is this hydroxide-like species is going to attack that carbon, just like we had seen in the SN2 reactions where we did the same thing, right? The base catalyzed hydrolysis of our carbonyls. Okay? And what's then going to happen is we will bond the OH to this carbon. This is going to make it so this can fall apart much easier. Again, we have to go through the tetrahedral intermediate that is stabilized by the oxyanion pole, right? Okay, so the last portion is going to be the carbonyl is going to come back down, reestablish. When it does that, one of these oxygens has to fall apart, right? Okay. If this oxygen falls off, then we go back this way. If the blue one does. If the black one does, we go forward and we have now released our product, right? There we go. Okay. Now we actually have one more step that has to happen to reset this and actually get to the place where we could do these reactions, okay? So we've released this, we no longer have the acyl enzyme, we have the free enzyme now, right? Okay. But in this case, we have the serine here, we have the carboxylate, and we still have this sort of stuck in the region, right? So what actually has to happen next is we actually have to have the remaining protein portion get the hell out of the way, right? And so the next step is we get it the hell out of the way, right? Which is this right here. Then what's going to happen on this next step is we're going to have a new protein come in and we do all this again. Okay? And that's all that is. This is coming into the area so that we can start doing the rest of the bonding. Okay? Okay. Does anybody have any questions about this process? Nope. Fantastic. Okay, remember that this is, though this technically is the chemotrypsin I was talking about with the specific numberings and such we'd mentioned, but this is also the catalytic triad as a more broad mechanism. Okay? They're the exact same thing. Okay, this is actually the oxyanion hole right here. This is what this looks like. So this is a region of the active site that will stabilize your normal unstable tetrahedral reactive intermediates. This will also help the transition state, so that precedes the formation of your tetrahedral intermediate. And so before you form the tetrahedral intermediate, this state right here, this is still going to be hydrogen bonded in there, and it's going to make it easier for the electrons to get pulled up onto that oxygen, right? So it actually helps the transition state before you hit this intermediate here as well, okay? That state where you're actually transferring electrons. It makes that carbon a little bit more positive than it would normally be, right? Okay, this is how it does it, by the way. So here we can actually see the, again, the oxyanion hole. That's that right there. See how it comes up here and incorporates the oxygen. Okay. And notice this is the serine right here that's going to do the covalent attachment to the carbon in the substrate. Okay? Everybody can see that okay? Okay. So this is that serine right here. Right there. We're covalently bonded to this one. Our oxygen comes up right here. The next amino acid right here is actually going to hydrogen bond to this. Everybody see that okay? Fantastic. Note this involves, right here again is our serine. Right there's the oxygen for it, the nitrogen. This is going to be 194, 193, right? Okay, and we have two hydrogen bonding sites. These hydrogen bonding sites are actually going to take a little bit of that electron density away from that alkoxide ion, right? Okay. Anybody have any questions about that? Don't lose anybody anywhere? Okay. Okay, it's actually kind of important you understand that because this is our first time we're actually seeing how an active site works, okay? So if you have any questions about how the hell to look at these things, now's the time to get them out of the way, right? Because we are going to see that more. So the S1 pocket, the specificity of the pocket of the chymotrypsin is actually going to favor binding the residues with long hydrophobic side chains. This is the selectivity or the specificity, correctly said, of this chymotrypsin. Remember how when we looked at chymotrypsin, it only cleaves after certain amino acids, right? Okay. This is how that's being specified, the specificity of it. Okay, so the S1 pocket, this is going to be a deep hydrophobic pocket. We're actually seeing it right here, into which the long on-charge side chains can fit. The side chain binding positions, the adjacent peptide bond for your cleavage. It's a cis-cell bond, that's the bond to be cleaved, is that right here. See that? That's that one. Okay? That's the cis-cell bond, right? I.e. scissor bond. Okay? Same root word. Okay. And so if you were looking at this, the amino acid that precedes that bond is going to have a what sort of side chain? Long hydrophobic side chain, right? Is it going to have a highly charged side chain? No. If it had a hydrophobic side chain but is not very long, it's going to have a little bit more trouble bonding. But theoretically, if you had something that was similar to this, you could do it, okay? However, there's only very limited ones in the actual natural amino acids that could do it. Right? Synthetically, though, you can make it. Right. So the residues on the amino terminal side of the cis-cell bond will be labeled the P1, P2, P3, and so forth. The residues on the carboxyl side of the cis-cell bond will be labeled P1, P2, P3 with primes. You always see these primes as being the next set of something in biochemistry, okay? So when you see a prime, it's a set of something that you have done some modification to, right? So for example, on this case, since we typically refer to the amine side as the leading edge, right? And with the carboxyl side would be the ending. The P1, P2, P3, and so forth would be the amine side. The other side that we're going to cleave, the carboxylate side, is going to have the second set of these. P1 prime, P2 prime, P3 prime, okay? P4 prime, etc. Okay, the corresponding sites on the enzyme are actually going to be labeled S1, S2, or S1 prime, the S2 prime. These are the sites on the enzyme that would bind to those, okay? So your P3 site, your P3 on the protein that you are going to be cleaving is going to line up with what? S3, right? The P2 with S2. S2 being on the enzyme, P2 being on the protein to be cleaved, right? That's going to be on the amino side, okay? On the side that comes after the carboxylate, on the side this side of the carboxyl side of the cis-cell bond, we are going to end up having the S1 prime, which is on the enzyme. That's going to match with the P1 prime, right? These will be the binding sites that will help to specify and give you the specificity. By the way, the S substrate bonding or specificity, right? Both work for S. The terminology is actually quite nice here. P for the protein that's going to be cleaved, right? Yeah, somebody actually thought about this. So again, the calic triads are going to be found in other hydrolytic enzymes. This is very common for hydrolytic splitting. If you're going to do a hydrolytic splitting, it's going to have one of the most common ones is calic triads. Okay? There are other ones that can do hydrolytic splitting, but this is one of the most common. So trypsin elastase are going to be homologs of the chymotrypsin. They're going to have different substrate specificity, but otherwise that active site is pretty much the same. Okay? The difference is going to be due to small structural differences in your S1 pockets. This being the S1 pocket. See that one right there? That being the one that it splits after this amino acid. That would specify the amino acid it splits after, right? Because it's the closest to the cis-cell that leads in front of it. Okay? Okay. Other proteases are not going to be homologs of the chymotrypsin. They'll contain similar active sites because these sites will have evolved independently. There's several different of these ones that do hydrolysis reactions or hydrolases that are going to be independently evolved. They have some similarity, but they're not homologs. They're not the same active sites with the same side chains in them. Okay? But these set are the exact same. This right here is the structural similarity of trypsin and chymotrypsin. And as you can see, the red and blue are the trypsin and chymotrypsin. And there is very little difference in these, isn't there? What's the difference? The big difference is the S1 site, right? And so if you look at this right here, the chymotrypsin has a deeper S1 site that's hydrophobic. The trypsin then has a, see how it has a negative charge group in there? What do you expect this to bind to? Something positive charge, right? Yeah. How about this one? This is the elastase. What's the difference between this and chymotrypsin? This is a deep hydrophobic pocket. Is this deep anymore? Shallow. Yeah. This one has a shallow hydrophobic pocket. Very good. Remember, again, these are the S1 pockets. These are the ones that are right before your cis-sil bond, right? The one you're going to be cleaving. That's how that selects. This, again, is the oxyanion hole. So here we see the calic triad and the oxyanion hole. This is for subtilissin. And that one's a little bit different, as you can see here. The previous one, it was basically 1, 2, 3, 4, right? This one, it actually wraps back around. You have a 70 or so difference in the amino acid count, okay? So it's actually not composed of a straight chain that's wrapping around. It's composed of something that wrapped around later. All right. Notice, by the way, though, the serine still is involved in the oxyanion hole because it's so close to. The oxyanion hole itself has to be so close to the site, right? So you still end up with a serine that's involved in it, okay? It just, the other half of that wraps around, 70 amino acids down. All right. Okay, so scientists have dissected the calic triad. We've used site-directed mutagenesis. They have used site-directed mutagenesis. God. The mutating residues of the calic triad of subticillin are to the alanine shows the scientist, allows the scientist, to quantify the importance of each residue for calic activity. This has resulted in dramatic loss of activity, has little impact on the KM on the other hand. So this mutation of the calic triad, we can actually see that this mutation to alanine gave you a lot less product coming through, right? But our binding didn't change significantly, okay? All right, so the results support the notion the calic triad acts to generate a powerful nucleophile to attack the carbonyl carbon atom of a peptide bond. The mutated enzymes still hydrolyze peptides faster than buffer, so they still have calic activity due to the fact that they have arrangement calic activity, but they have lost an important portion of that calic activity due to the loss of that especially modulated base that we talked about, right? Okay, and so this is actually the site-directed mutagenesis of the subtilissin. God, I didn't get my tea. And it shows. Okay, so this right here, remember whenever you do an activity or binding diagrams, do them in logs for God's sake. I tell you what, this entire COVID era shit has just driven me insane with them going 30% increase in binding. 30% increase in binding is nothing, right? That's ridiculous. You do a log, okay? Logarithmic. So this is the log of the kcat here. Right here we see the wild type, and that's the one that has the most calic activity, right? And so we've done some various mutations. So we have changed the S221A, H64A, etc. So we changed different sites, what amino acids will be expressed, okay? And if we see a significant drops in the calic activity, then we know, well, that change was meaningful, right? Now, if you change something that didn't have a meaningful activity, could you see a drop, a change? Yeah. You could change something structural, and it changes that cleft just slightly, right? If it changes the cleft just slightly, we might see a 30%, 60% drop, okay? But when you're looking at binding, binding is a logarithmic activity, right? When you're looking at catalysis is a logarithmic activity. So the difference is in activity between this and this form. This is a, this is 1, 2, this is -5, right? So that is a 10 to the -7 difference, right? That's 10 to million times difference, right? Okay. And so that's the sort of calic activity difference you're going to see if you actually replace something in the binding site, or that's actually involved in that binding, or involved in the actual calic activity, okay? You won't see a, like, 10 times difference, okay? Yeah. Does that make sense? All right. Now, note that all of these forms, even though we have disturbed that reactive site on the enzyme, and we've caused changes in it that have severely affected its ability to do this reaction, we have actually still have some calic activity above and beyond the on-catalyzed enzyme. Something like a 10,000, 100,000 times difference, right? So just the binding to it, even though we don't have all the other stuff, makes a difference. All right, so cysteine proteases, these are going to rely on a cis residue. These are going to be activated by the his residue. They're going to play the role of a nucleophile that attacks the peptide bond. Aspartyl proteases, these will use a pair of ASP residues. This is kind of a unique one because it actually uses a pair of the same. That's going to act together. This will allow a water molecule to attack the peptide bond. This one's kind of interesting because it's a much more the sort of approach we would take as humans, where we activate some sort of water or small molecule to attack. Metalloproteases, this will use a bound metal ion. The most common bound metal ion for this is zinc. This is going to activate the water molecule to act as a nucleophile to attack the peptide carbonyl bond. This sounds like something we would have done in organic chemistry, doesn't it? Yeah. This is what these look like, by the way. So in each of the 3 classes of enzymes, the active site is going to include features that are going to act to activate a water molecule, in these cases here, or another nucleophile. This is the other nucleophile case right there. This will polarize the peptide carbonyl group right here, making it more reactive. It will also stabilize the tetrahedral intermediate. Okay? So they're very similar. If you were to look at them from an organic chemistry perspective, you would go, that's pretty much the same thing, right? But from a biological perspective, they're different because they're different amino acids, right? Okay. Remember, these are classes of proteases. I'm not talking about a specific enzyme at this point, okay? Okay, so this is, again, the cysteine. See the cysteine right here? We'll activate that cysteine so it becomes a more reactive intermediate, and we'll actually have attack by the cysteine on our carbonyl. We'll stabilize that alkoxide, and then go through the reaction, okay? Notice these are all basically like an SN2 reaction, aren't they? Okay? Yeah. Or more specifically, correctly stated, they're an addition-elimination reaction, right? This right here is the aspartyl protease. This one's kind of cool because you have the same group twice, and both of those groups act a little bit differently. Okay? And it does have directionality, by the way, because your site has directionality. So there's actually a difference. So right here, what we're going to have is we have the activation of a water. This will make it more base-like, more like a hydroxyl, right? And then this is then going to attack this carbonyl, and then this will stabilize this alkoxide intermediate. This one stabilizes it quite differently than what we've seen before. See that? Okay, so this is the metalloproteases. This is one of the reasons why you need zinc in your diet, by the way. If you ever wondered, why the hell do I need to eat zinc? There you go. Okay, there's one of the reasons. And this one's pretty cool because this is a base right here. This has a very low, this can be many different basic side chains. We don't really care. Okay, anything that has basicity. And so what's going to happen is you have as binded zinc. Notice we don't even specify what's binding to zinc. Okay? And that zinc will have a 2+ charge and 1 coordination site that's free here. And so the water will actually attach to this. Remember when we talked about the acids back in the day, Arrhenius acids? And we're like, metal ions can make things more acidic. Remember that? Yeah. That's what we're doing here. We're attaching this to a zinc, making it more acidic. It loses the proton to the base, and this gives us a stabilized hydroxyl, right? Cool, huh? We've tied all the way back to like chapter 1 in Gen Chem. All right, so this is the protease inhibitors. These are going to be drugs that block the protease activity. Captopril is going to be an inhibition of the metalloproteases. Angiotensin converting enzyme, which is ACE. This will actually help to regulate blood pressure, in case you're wondering. This right here is Enadrir. I forget which is the brand name. I think this is the brand name, if I remember correctly, which is Crixivan. This is actually an inhibitor of HIV protease. This is an aspartyl protease. Remember the aspartyl protease is up here? There. And that usually would cleave the multi-domain viral proteins into their active forms. We'll reduce that activity. We'll get less of the viral proteins in their active forms, and thus it's going to reduce the deaths due to AIDS because it doesn't develop as fast, right? This is designed to resemble a peptide substrate of the enzyme, and so this would be like a competitive inhibitor, right? Though, keep in mind, these actually do covalent modifications on them and covalently bind, right? This right here is that HIV protease. It has twofold symmetry here. And so what we can see here is this is Endovir. This is the drug binding into our HIV protease that you see this twofold symmetry. Okay? Carbonic anhydrases make a fast reaction that we would normally consider a pretty fast reaction, even faster, okay? And really, really fast. So carbon dioxide being our end product of our aerobic metabolism, did anybody here think that carbon dioxide was transported backwards by your hemoglobin? You'd all been thoroughly disabused about that, right? Okay. So how is carbon dioxide transported in your blood? As carbonic acid, right? Okay. And so this is how this is going to be done. So we're going to take that CO2 in our blood after we do the end product of aerobic metabolism. We're then going to react it with the water in our blood. This is going to make bicarbonate ion, which will then make carbonic acid. Okay? So ultimately, this is what's going to happen in our blood, red blood cells. We will then transport this carbonic acid, which is water soluble, through our blood to our lungs. At our lungs, we will do the reverse process, and the carbonic acid is going to go back to CO2, which we then breathe out, right? And this is how we get rid of CO2. Does anybody have any questions about that? Okay. This process right here is the carbonic anhydrases. So they'll convert the CO2 right here into the bicarbonate ion, which is already somewhat soluble. This also does act to increase the acidity, by the way. This is actually the major buffer system in our blood, in case you're wondering. Okay? Bicarbonate carbonic acid is the major buffer system in our blood. And that's why we have a pKa of 7.4. That's why pH is 7.4 for physiological systems, in case you're wondering. So then next, it's going to dehydrate the bicarbonate ion in the blood to form CO2. This will happen at the lungs. Again, an enzyme goes both directions, right? Okay, so the carbonic anhydrases is going to contain a bound zinc ion essential for the catalytic activity. The human genome contains at least 7 carbonic anhydrases with its own gene. Yeah, there's 7 different copies of this in the human genome. All are homologous, with substantial sequence identity. The zinc 2+ is going to be bound to 4 ligands. The imidazole rings of 3 his residues, and the water molecule or hydroxide ion, depending on your pH in that region. Those will be the 4 things that are bound to the CN2+. Right here we see the active site of the human carbonic anhydrase, 2 in this case. That gives you the full protein. This actually includes the bound zinc ion right there, blown up, right there, small. Okay, and these are the 3 his residues right here. That's the water that will end up being stabilized, so it will donate its proton more easily. Okay, and become more like a hydroxyl, will be stabilized. Okay, so at pH 8, the reaction actually is going to proceed near its maximal rate. So as your pH decreases, the rate of the reaction will drop. The midpoint is actually near pH 7. So this suggests a group loses a proton at pH 7. And so it has about a pKa of 7. And so you notice this all happens around 7 to 8. What is our blood? 0.4, yeah, 7.4, which is about halfway between those, right? What's helping to modulate that? This is one of those key factors. Okay. Anyways, this is the catalytic activity. This is the pHs. Here you can see the pH 7, and here's 8, where it starts losing activity. Okay? Fantastic. Again, the zinc 2+ is going to lower the pK of water from the 15.7 that's normal. By the way, that's what makes that pH of 14 scale in water. So the pK of water, normally 15.7, gets lowered down to 7 due to it binding this zinc 2+, meaning it's a much more likely to lose those protons, right? And form the hydroxyls, okay? Or the hydroxide, I should say in this case. Okay. So again, this is that reaction. Notice the binding of the zinc is stabilizing that oxide right there. Okay, so we have this carbonic anhydrase has a CO2 binding site that's adjacent to our active site on the zinc ion. It needs to be hydrophobic because CO2 is what? Hydrophilic or hydrophobic? Hydrophobic. CO2 is hydrophobic. It has no dipole. And so if we had this highly charged site, we wouldn't have CO2s coming into it, right? And we'd have a large binding problem, even though our reaction rate would be pretty good for whatever we bind, right? Yeah. And so right next to this site, which again, if we look at the overall diagram is right here, we'll have a CO2 binding site. That CO2 binding site will have a lot of hydrophobic groups. So we'll have a hydrophobic patch. This will bring the CO2 in close proximity. And so you can see here, this is the zinc right here. Everybody see that okay? Okay. Then we have the water molecule associated with it. And then we have the hydrophobic patch over here where the CO2s will bind. And they'll sort of vibrate and move around on that, right? And come in contact with our reactive groups. Okay? Can everybody envision that? Yeah. Nice, right? Yeah. It even orients it nicely. See how it's oriented? The hydrophobic patch is oriented in such a way the carbon would be exposed to our binding, right? Yeah. It's very nice. So the binding of the water to the zinc ion actually favors the formation of the transition state. This does it by facilitating the proton release from the water, making the hydroxyl, or excuse me, I keep saying hydroxyl, because I'm so into protein right now mode, and serine. Hydroxide, excuse me, it positions the water molecule to be in close proximity to the other reactant, which is our CO2, and in the proper orientation, which is the oxygen being in close proximity to the carbon, right? Okay, and so it actually is a fairly small cyclic reaction here. So we have the water binding the zinc 2+. This is going to release H+ into the blood. This is then going to give us the hydroxide ion. Next, we're going to have the CO2 that binds to that hydrophobic site through dispersion forces, of course, and exclusion from the water. This is going to orient that in the correct direction and allow that to come in contact with the hydroxide, right? Again, we have this very reactive carbon. The carbon would have a positive charge, right? It's just that it doesn't have an overall dipole because it's symmetric, okay? And so these electrons will flow into that carbon right here, forming a bond, and then the electron in one of these oxygen-carbon double bonds that are delocalized would flow over, forming the alkoxide. Now notice that alkoxide is actually stabilized by the zinc 2+, which can attract that electron density over and stabilize it, okay? So what will happen next is we'll actually get this step now. That's going to be where we have the, we now have the carbonic acid right here. This is going to come off, detach, and be replaced by an H2O. This typically happens due to the H2O literally coming in and hitting these all the time, and the energy favoring the water attaching to this, and then the carbonic acid coming off and making hydrogen bonds to all the waters, right? Remember, that is an ionic dipole interaction going on. Kind of the same process as you would get if you, this association in water. So the zinc-bound water molecule must lose a proton to regenerate the active form of the carbonic anhydrase. The equilibrium constant is going to be K equals 10 to the -7 molarity. And that K, of course, is the K1 over the K minus 1, as we saw before, which is that binding, right? Okay, so the K minus 1 is going to be limited by the rate of proton diffusion. This is going to have a second order rate constant of approximately 10 to the 11th, molarity to the -1, second to the -1. Everybody remembers diffusion fine. Remember that things don't straight off shoot where they're supposed to go. What they have to do is wander, right? And this is sort of the maximum diffusion you could get. Okay? Okay, so the K1 must be less than equal to the 10 to the -4 seconds to the -1. This is because the K1 has to equal this K value times K to the -1, right? Okay, and so this is the overall process we're looking at here. One of the things that has to happen here is this H+ actually has to pop off and leave, right? The H+ doesn't leave, we can't get the reaction with the carbon dioxide, okay? Okay, and that's the K value right there, 10 to the -7. Let's talk about some of the roles of buffers. So the highest rate of CO2 hydration, what's my time? It's past? Okay. Okay. Oh, well, you all have a wonderful weekend. I'll stop here, and man, I really slowed down today, didn't I? Damn.